Good day, everyone. Welcome to NBio, or shall I say, welcome back. Today, we're diving into lesson 3, all about biomolecules. This topic is foundational not just in biology but across disciplines like agriculture, forestry, health sciences, and biotechnology. So, before we explore the details, let's clarify what you're expected to learn from this lesson. These outcomes will guide our discussions and help you focus on essential concepts. Learning outcome number one: first, you should be able to identify the major categories of biomolecules. So, this include carbohydrates, lipids, proteins, and nucleic acids. Each plays a distinct role in living systems and will explore their characteristics in depth. Learning outcome number two: you learn to distinguish between the structures and functions of these biomolecules. So, understanding how their molecular architecture influences their biological roles is key to grasping how life operates at the cellular level. And third, you'll be able to explain their relevance in health, agriculture, forestry, and biotechnology. Whether it's nutrient formulation for crops, genetic engineering in plants and trees, or disease diagnostics, biomolecules are at the heart of innovation and sustainability. So let's begin our exploration of biomolecules by asking a simple question. What are biomolecules and why do they matter? Biomolecules are organic compounds produced by living organisms. They are built primarily from five key components such as carbon, hydrogen, oxygen, nitrogen, and phosphorus. These elements form the molecular backbone of life, shaping everything from cellular structure to metabolic function. There are four major categories of biomolecules, each with distinct roles in biological systems. The first one is carbohydrates. Carbohydrates are your primary energy source. They also provide structural supports in plants, just like cellulose in cell walls. Next one, we have lipids. Lipids are often misunderstood as just fats. In fact, lipids are crucial for long-term energy storage and insulation. They also form cell membranes which regulate what enters and exits the cell. The third biomolecule we have here is proteins. So proteins are the workhorses of the cell. They act as enzymes, transport molecules, and provide structural integrity. Their diversity in function stems from their complex and variable structures. The last one we have is nucleic acids. So these include DNA and RNA. So the molecules are responsible for storing and transmitting genetic information. They are the blueprint guiding cellular processes and inheritance. Now that we have introduced the four major biomolecules, let's begin with the first category, carbohydrates. These are among the most abundant organic compounds in nature, and they play a central role in both energy metabolism and structural integrity, especially in plants. Carbohydrates follow a general formula of CH2O, so enclosed and open and closed parenthesis sub n, where n is equal to any number. So this general formula reflects their composition of carbon, hydrogen, and oxygen in a 1:2:1 ratio. Their basic building blocks are called monosaccharides. So monosaccharides are simple sugars like glucose and fructose. So let me just repeat that. The building blocks of carbohydrates are called monosaccharides. When monosaccharides link together, they form larger carbohydrates through glycosidic bonds. These covalent linkages determine the structure and digestibility of the resulting compound. Carbohydrates are classified based on the number of sugar units they contain. The first classification is monosaccharides. Monosaccharides are single sugar units. Mono means one or single, saccharides mean sugar. So they are single sugar units. For example, glucose fuels cellular respiration. So glucose is a monosaccharide which fuels cellular respiration. Then we have disaccharides. Di means two, saccharides means sugar. Or meaning, or in other words, disaccharides consist of two monosaccharides joined together. So this is a common example, sucrose or table sugar. So sucrose is a disaccharide. Then don't forget we also have oligosaccharide consisting of 3 to 10 monosaccharides joined together. Okay, don't forget oligosaccharide. Then we have polysaccharide, meaning to say these are long chains of sugar units. So poly means many, saccharide means sugar. So of course, they form long chain of sugar units. Examples of polysaccharides include starch. So starch stores energy in plant, while cellulose, another polysaccharide, provides structural support in cell walls. Now, let's take a closer look at glucose, the most well-known monosaccharide and a central molecule in energy metabolism. Glucose exists in two structural forms, linear and cyclic. The linear form, shown here on the left side, presents glucose as a straight chain of carbon atoms, each bonded to hydrogen and hydroxyl groups. This form is useful for understanding how glucose reacts chemically, especially during synthesis and breakdown. However, in aqueous environments like the cytoplasm, glucose predominantly adopts a cyclic structure, as shown on the right side. This ring form is more stable and biologically active. It's the version that participates in most metabolic pathways, including glycolysis and starch synthesis. Why does this matter? Understanding the structures help us grasp how glucose interacts with enzymes. So enzymes, and then this also forms glycosidic bonds and contributes to larger carbohydrates like starch and cellulose. In agriculture and forestry, this knowledge informs how we evaluate crop quality, post-harvest sugar dynamics, and even fermentation processes in biotechnology. Okay, let's now shift our focus to starch. Starch is one of the most important polysaccharides in both plant physiology and agricultural science. Specifically, we look at amylose, which is a major component of starch. Amylose is composed of repeating glucose units with the formula or molecular formula C6H10O5. These units are linked by alpha 1,4 glycosidic bonds, forming a long and branched chain. Typically, amylose contains between 300 to 600 glucose units, depending on the plant source and developmental stage. So the linear structure of amylose allows it to coil into a helical shape as shown in the 3D molecular model. And this conformation influences its digestibility, gelatinization behavior, and interaction with other molecules, especially in food processing and post-harvest applications. Now that we have explored the structure of carbohydrates from glucose to amylose, let's connect that knowledge to their biological roles and agricultural applications. Carbohydrates serve three essential functions in living organisms. First, it serves as the primary energy source. So, for example, Glucose fuels cellular respiration through glycolysis, providing immediate energy for metabolic processes. The second one, we have structural support. For example, cellulose. Cellulose is a polysaccharide which reinforces plant cell walls, contributing to rigidity and protection. The third biological role of carbohydrates is energy storage. For example, glycogen stores energy in animals, while starch performs the same role in plants. So the stored energy in animals is called glycogen, and the stored energy in plants is called starch. These reserves are mobilized during periods of high energy demand or stress. In agriculture, carbohydrate content directly influences crop quality and utility. For example, we have starch content affects the texture, sweetness, and drying behavior of crops like sweet corn. High starch levels may improve shelf life, but it may reduce palatability if not managed properly. Next one, we have cellulose. Cellulose impacts forage digestibility. In livestock nutrition, understanding cellulose levels helps optimize feed formulations for better nutrient absorption and animal performance. Let's now turn our attention to lipids. Lipids is another essential class of biomolecules. While often associated with fats, lipids encompass a diverse group of compounds with critical roles in energy storage, insulation, and cellular architecture. Okay, so what are the basic types or what are the basic units and bond type of lipids? So we have here lipids are composed of fatty acids and glycerol. These components are joined by ester linkages. Okay, remember ha, kung ang carbohydrates ganina, they are formed through glycosidic linkages. Lipids, on the other hand, are joined by ester linkages. Okay, so this forms structures like triglycerides and phospholipids. Unlike carbohydrates, lipids are not built from repeating monomers, which gives them structural flexibility and functional diversity. There are three major types you should be familiar with. Triglycerides, these serve as long-term energy reserves. In animals and seeds, they provide dense energy storage. Then we have phospholipids. These form the bilayer of cell membranes, regulating transport and maintaining cellular integrity. Steroids. These function as signaling molecules, including hormones like testosterone and estrogen, which regulate physiological processes. So by the time that you will enroll general biochemistry, you will learn that there are actually multiple types of lipids, but this one, triglycerides, phospholipids, and steroids, are just the three major ones. Okay, so it will be more complicated in general biochemistry. Then we have lipids can be classified based on saturation. For example, we have saturated fats contain no double bonds, so making them solid at room temperature. So these are very common in animal fat. So kabantay mo ng mantika sa baboy, di ba? Usahay pakupsan nato ang baboy kung maghumba, tapos ato isat aside ang mantika, di ba? Pagkaugmaan na kay mura siya mo solidify at room temperature, okay? So meaning to say that is a saturated fat. Since there are no double bond present, walay double bond present, so this makes them solid at room temperature. Next one, we have unsaturated fats. Okay, they have one or more double bonds. So sa unsaturated fats na ay one or more double bonds. So this introduce kinks in the fatty acid chain, making them more fluid or found in plant oils and fish oils. Okay, let's proceed. Now that we've discussed the basic types of lipids, let's zoom in on one of their most critical roles. forming the plasma membrane. This structure is fundamental to all living cells, and its integrity depends heavily on the lipid composition. We have here structure of the plasma membrane showing the phospholipid bilayer. At the heart of the membrane is the phospholipid bilayer. Each phospholipid has a hydrophilic head and hydrophobic tails, allowing it to self-assemble into a double layer that separates the internal environment of the cell from the external surroundings. But the membrane isn't just lipids, it's a dynamic mosaic of molecules. So for example, integral and peripheral proteins regulate transport, signal reception, and structural support. Then we also have glycoproteins and glycolipids. They play roles in cell recognition and immune response. Then we also have cholesterol. Cholesterol modulates membrane fluidity, especially under temperature stress. Protein channels allow selective movement of ions and molecules across the membrane. Okay, let's move on to the next slide. This is a diagram of a phospholipid bilayer. So, let's take a closer look of the structure of phospholipids, which is known to be the building blocks of the plasma membrane. Their unique chemical properties allow them to form the bilayer that surrounds all cells. Phospholipids are known to be amphipathic molecules. Amphipathic means they have both hydrophilic and hydrophobic regions. The hydrophilic head is composed of a phosphate group and glycerol, which interacts favorably with water. On the other hand, the hydrophobic tails, made of fatty acids, repel water and prefer to interact with other lipids. Meaning to say, hydrophilic head is water-loving, while hydrophobic tails are water-fearing. Ano water-fearing man ang hydrophobic tails? Because they are only made of fatty acids and therefore they repel water and prefer to interact with other lipids. Okay, so in terms of saturated versus unsaturated tails, one tail is typically saturated, meaning it has no double bonds and remains straight. The other is unsaturated, containing one or more double bonds that introduce bends in the chain. These structural differences affect membrane fluidity and permeability. When placed in water, phospholipids spontaneously arrange into a bilayer with hydrophobic tails facing inward and hydrophilic heads facing outward. This arrangement forms a semi-permeable barrier that regulates the movement of substances in and out of the cell. Okay, so if we're going back into this diagram, we could see that the hydrophobic tails facing inward, while the hydrophilic heads facing outward. Okay, so that's it. Now let's proceed to the next slide. Diagram of triglyceride molecule. So now let's take a closer look at triglycerides, the most common form of lipids found in both plants and animals. These molecules are central to long-term energy storage and thermal insulation. A triglyceride is composed of one glycerol molecule bonded to three fatty acid chains. The glycerol backbone contains 3 carbon atoms, each linked to a hydroxyl group. These hydroxyl groups form ester bonds with the carboxyl ends of the fatty acids, resulting in a compact, energy-dense molecule. Each fatty acid chain is a long hydrocarbon tail ending in a methyl group. The chains may vary in length and saturation, which influences the physical properties of the triglyceride, such as whether it's solid or liquid at room temperature. Triglycerides actually serve as energy reserves, especially in seeds and adipose tissue. They provide more than twice the energy per gram compared to carbohydrates, making them ideal for long-term storage. Let's now examine how triglycerides are formed at the molecular level. This reaction illustrates the synthesis of triglyceride, a type of lipid that plays a central role in energy storage. On the left, we see glycerol, a three-carbon molecule with hydroxyl groups attached to each carbon. This backbone will serve as the anchor for the fatty acid chains. Each fatty acid consists of a carboxyl group attached to a long hydrocarbon chain. When three fatty acids react with glycerol, they form ester bonds through a dehydration synthesis reaction, releasing water molecules in the process. The resulting molecule is a triester of glycerol, where each hydroxyl group has been replaced by an ester linkage to a fatty acid. This structure is compact, hydrophobic, and energy dense, ideal for long-term energy storage. Okay, let's proceed to the next slide. Now that we have explored lipid structures, from phospholipids to triglycerides, let's connect those forms to their biological roles and agricultural relevance. Okay, lipids perform several vital functions in living organisms. We have long-term energy storage, which means triglycerides store energy efficiently, providing more than twice the energy per gram compared to carbohydrates. Then we have thermal insulation and protection, which means in animals, lipids cushion organs and help regulate body temperature. Then we also have hormone synthesis. So steroids like testosterone and estrogen are derived from lipids and regulate growth, reproduction, and metabolism. The last one we have is membrane fluidity and permeability. Phospholipids and cholesterol maintain the dynamic nature of cell membranes, influencing the transport and signaling. In terms of agricultural relevance, so we have in agriculture, lipid content directly impacts productivity and quality. For example, we have feed energy value, wherein lipid-rich feeds offer higher caloric density, improving growth rates and feed efficiency in livestock. Then we have seed viability and germination, wherein phospholipids are essential for maintaining membrane integrity during seed storage and early growth stages. Their composition can influence germination success and seedling vigor. Let's now explore proteins. Arguably the most functional, diverse biomolecules in living systems. So from enzymes to structural components, proteins are involved in nearly every biological process. Proteins are made up of amino acids. There are 20 standard types, each with a unique side chain that determines its chemical behavior. These 20 amino acids are linked by peptide bonds. So I hope you can still remember the bonds found in carbohydrates are glycosidic linkages or glycosidic bonds. In lipids, we have ester linkages or ester bonds. Then in proteins, we have peptide bonds. Okay? These amino acids are linked by peptide bonds, forming long chains called polypeptides. So to understand how proteins function, we need to examine their 4 structural levels. We have primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure. This is the linear sequence of amino acids, like beads on a string, so the order determines everything that follows. Secondary structure, so it looks like local folding patterns such as alpha helices and beta sheets. So they are stabilized by hydrogen bonds. Then we have tertiary structure. This is the three-dimensional folding of the entire polypeptide driven by interactions among side chains. So this level determines the protein's functional shape. Then we have the quaternary structure. Some proteins consist of multiple polypeptide chains working together, like hemoglobin, which carries oxygen in the blood. So a protein's function is directly tied to its structure. Even a single amino acid change can disrupt folding and lead to loss of function, or it may cause a disease. So to elaborate, let's now visualize the four levels of protein structure, each one building upon the last to create a functional biomolecule. Primary structure. This is the linear sequence of amino acids like beads on a string, and it is determined by the genetic code and sets the foundation for all higher order folding. The secondary structure, here, the chain begins to fold locally into alpha helices and beta sheets, so stabilized by hydrogen bonds. These patterns give proteins their initial shape and mechanical properties. Tertiary structure, this level represents the three-dimensional folding of the entire polypeptide. It is driven by interactions among side chains, such as hydrophobic forces, ionic bonds, and disulfide bridges. This is where the protein takes on its functional form. Then the last is the quaternary structure. Some proteins consist of multiple polypeptide chains that assemble into a large complex. So hemoglobin is a classic example, with four subunits working together to support oxygen. Okay, let's move on to the next slide. We have here a visually consolidated diagram of our primary structure, secondary structure, tertiary structure, and quaternary structure of our proteins. So in the primary structure, of course, I have said it before that we can see a linear chain of amino acids. And then in the secondary structure, the chain begins to fold locally into alpha helices and beta sheets. And the tertiary structure shows the three-dimensional folding of a single polypeptide chain. And then finally, quaternary structure consists of multiple folded polypeptides assembled into a functional complex. Okay, next slide. We have the functions and applications of proteins. So let's connect that complexity to their biological systems and agricultural practice. So first, we have proteins are incredibly versatile. So meaning their functions depend on their structure, and they serve in nearly every cellular process. For example, we have proteins also acts as enzymes. So meaning biological catalysts like amylase and rubisco accelerate the reactions from digestion to photosynthesis. Then we also have structural proteins. So molecules like collagen and keratin provide strength and resilience to tissues. Then we also have transport proteins like hemoglobin, for example, carries oxygen through the bloodstream. And last, we also have the biological role of proteins. So it serves as immune defense. We have antibodies. So antibodies recognize and neutralize pathogens, forming the basis of immune protection. So in agriculture, proteins are central to productivity and innovation. So enzymes and post-harvest processing improve food texture, shelf life, and nutrient availability. Next, we have protein content in feed directly affects growth rates, reproductive success, and overall livestock health. Then we also have genetic engineering. So using recombinant proteins to enhance crop resistance, produce vaccines, and develop biofortified foods. Okay, then let's have the last biomolecule, the nucleic acids. So nucleic acids are composed of nucleotides, each made up of three components. So we have a sugar, a phosphate group, and a nitrogenous base. So by the way, nucleic acids are the molecules which serves as blueprint of life because they store and transmit genetic information across generations and guiding cellular function. So again, let me repeat, nucleic acids are composed of nucleotides, and then each of the nucleotides are made up of three components. Number 1, sugar, then number 2, phosphate group, and number 3, nitrogenous base. So these nucleotides are linked by phosphodiester bonds. Okay, so I hope you can still recall sa carbohydrate, iyang bond kay glycosidic linkages or glycosidic bond. Then sa lipids, we have ester linkages or ester bonds. And then sa proteins, we have peptide bonds. And then finally, sa nucleic acids, we have phosphodiester bonds. So these nucleotides are linked by phosphodiester bonds, forming long chains that encode biological instructions. So there are two types of nucleic acids that you need to know, and I know you're very familiar with this, DNA, or known as deoxyribonucleic acid. So this is a double-stranded and stores the genetic code. It is responsible for heredity and cellular identity. Next, we have RNA, or ribonucleic acid. This is single-stranded and plays a key role in protein synthesis, acting as a messenger, translator, and regulator. Okay, so we have here a picture or diagram of DNA structure. So DNA, or deoxyribonucleic acid, is the molecule that carries genetic instructions in all living organisms. Each DNA strand is made of repeating units called nucleotides. So nucleotides are the building blocks of nucleic acids, okay? And this consists of phosphate group, a sugar called deoxyribose, okay? Ang sugar sa DNA is called deoxyribose. And a nitrogenous base, so it's either adenine, thymine, okay, adenine for A, thymine for T, cytosine for C, or guanine for G. These bases pair specifically, so adenine will pair with thymine, and cytosine will pair with guanine, so forming the rings of the DNA ladder. So the sides of the ladder are made of alternating sugar and phosphate groups, creating the sugar-phosphate backbone. So together, the two strands twist into a double helix, which is the iconic shape of DNA. This structure is essential for storing genetic information, copying it during cell division, and passing it on to the next generation. Okay, so let me just repeat the components or the nucleotides present in DNA. So we have here the nucleotides consist of phosphate group, a sugar called deoxyribose, and a nitrogenous base. So the nitrogenous base are either adenine, thymine, cytosine, or guanine. Okay, so just like what I have mentioned before, DNA is structured as a double helix, meaning two strands twist around each other like a spiral staircase. Each strand is made of a sugar-phosphate backbone between them and nitrogenous bases adenine, thymine, cytosine, and guanine. So they pair specifically. In DNA, adenine will pair with thymine and cytosine will pair with guanine. and they are held together by hydrogen bonds. These strands run in opposite directions, one from 5' end to 3' end, and the other from 3' end to 5' end, which is called antiparallel orientation. This arrangement is essential for DNA replication and accurate transmission of genetic information. Then, let's have the RNA structure. RNA, or ribonucleic acid, is typically single-stranded and plays a central role in protein synthesis. Each RNA nucleotide is made of phosphate group, a ribose sugar, and a nitrogenous base. Unlike DNA, RNA contains uracil instead of thymine. So, walay thymine ang RNA, but naa siya'y uracil. And uracil will pair with adenine during transcription. The presence of ribose with an extra hydroxyl group makes RNA more reactive and less stable than DNA. This is ideal for short-term tasks like carrying genetic instructions, assembling proteins, and regulate gene expression. Okay, so let us proceed. DNA and RNA are both nucleic acids. They differ in structure and function. So DNA is double-stranded and contains thymine, while RNA is single-stranded and uses uracil instead of thymine. So again ha, ang RNA wala siya'y thymine pero naa siya'y uracil, and uracil will pair with adenine. DNA stores genetic information long-term, while RNA helps express that information, especially during protein synthesis. So another key difference is the sugar. Ang sugar sa DNA kay deoxyribose, so making it more stable, while ang sugar sa RNA is called ribose. So please don't forget, the sugar of DNA is called deoxyribose, while ang sugar sa RNA is called ribose. So ribose includes an extra hydroxyl group, making it more reactive and suitable for short-term tasks. Now let's move on to the function and applications of lipids. Nucleic acids. Nucleic acids are the molecular foundation of life, right? So each type plays a distinct role. For example, we have DNA. They act as the blueprint for cellular function and heredity. So meaning it stores genetic instructions that guide development, metabolism, and reproduction. And we also have RNA. They serve as messenger and translator. So converting DNA's code into proteins through transcription and translation. And then we also have ATP, or adenosine triphosphate. Although technically a nucleotide, functions as the energy currency of the cell, so powering biochemical reactions from muscle contraction to active transport. So in terms of agricultural relevance, nucleic acids are more than just molecules. They are actually tools for innovation. For example, we have DNA markers used in crop breeding to identify desirable traits like drought tolerance, disease resistant, or improved yield. Marker-assisted selection speeds up breeding programs and enhances precision. Then we also have RNA interference is a powerful method for pest control. By silencing specific genes in pests, RNA interference offers a targeted and environmentally friendly alternative to chemical pesticides. Then we also have genetic modification, which relies on manipulating DNA to introduce improved traits such as vitamin-enriched crops, faster-growing livestock, or plants that thrive in marginal soils. Okay, so we have a table comparing the four major classes of biomolecules. So let's just Recall, no, carbohydrates are actually made of monosaccharides. They are linked by glycosidic bonds. Then lipids are formed by fatty acids and glycerols, joined by ester linkages. Then proteins consist of amino acids. They are connected by peptide bonds. And then nucleic acids are composed of nucleotides. So they are also bonded through phosphodiester linkages. Okay, then in terms of function, carbohydrates, energy source, and structural support. For example, we have glucose and starch. Then function of lipids, they serve as energy storage, insulation, and signaling. For example, we have oil and cholesterol. Then the function of proteins, they act as enzyme, transport, and structural roles. So for example, we have hemoglobin and rubisco. Then the function of nucleic acid includes genetic coding and protein synthesis. So we have examples like DNA, RNA, and ATP. Then we have another diagram here. Okay, so each macromolecule has a distinct molecular architecture and building blocks. So just a review. So carbohydrates are chains of sugar, often forming ring structures like glucose. So their monomer is called monosaccharide. While ang lipids, they are composed of fatty acids and glycerol, often forming bilayers or compact of triglycerides. So the key unit here is the phospholipid. Tapos, ang proteins, they are folded chains of amino acids with complex 3D shape to determine their function. Then we have nucleic acids are chains of nucleotides, each containing a sugar, a phosphate, and nitrogenous base. So their function is to encode genetic information. Okay, so kamo na ibahala og basa ana ha. Then let's go to the last slide. So as we close this lesson, let's step beyond the molecular diagrams and explore how biomolecules shape the world around us. So these compounds aren't just confined to cells, they are also embedded in our daily lives and our food systems, our medicines, and technology. So in terms of nutrition, every time na mukhaon tano, we're actually interacting with biomolecules. So every time we eat, we're interacting with biomolecules. So carbohydrates, lipids, and proteins are macronutrients. And these macronutrients are essential for energy, growth, and repair. So understanding their structure helps us make informed choices about our diet, food processing, and nutritional interventions. Then, medicine. In healthcare, biomolecules are the foundation of innovation. So protein-based drugs like insulin and monoclonal antibodies treat chronic diseases. Then we also have DNA diagnostics allow for early detection of genetic disorder and personalized medicine. Then we have in agriculture, in the field of agriculture, biomolecules drive progress. For example, we have biofortification, which enhances the nutritional value of crops. Then we have GMOs, or genetically modified organisms, use DNA technology to improve yield, resistance, and shelf life. Then we also have feed formulation, which relies on protein and lipid profiles to optimize livestock performance. Then we have biotechnology. So biotechnology applications are, for example, we have enzyme-based detergents. So they use proteins to break down stains efficiently. So mo day na makakuha og buling ang mga powder na detergents kay naman day sila enzyme-based. Okay, so their detergents are derived from proteins. Kaya di ba ang proteins, they also act as enzymes. Then we have RNA vaccines, like those developed for COVID-19. They demonstrate how nucleic acids can be harnessed for global health solutions. So that concludes our lesson 3. So I hope sa next session, you will also be prepared, and please be guided. After this video, I will post an activity. So you really have to participate with the individual activity that will be posted later. But that will be done during face-to-face. Okay, that would be all. And thank you so much. See you tomorrow during our face-to-face class.